Hello everyone and welcome to today's session. And today we'll be discussing NCERT exercises for the chapter Human Health and Diseases. This is the disclaimer. So what are the objectives of today's session? So today we will understand the basic concepts related to the chapter 7 Human Health and Diseases by working through NCERT exercises. So these questions are based on some concepts. We'll discuss these concepts by working through NCERT exercise questions. Okay, so today we'll be discussing the NCERT exercises questions for the chapter Human Health and Diseases. Okay, so let's begin with the session today. So let's discuss this question. What are the various public health measures which you would suggest as safeguard against infectious diseases? For more information, you can read NCERT section 7.1. So let's have a look at the answer. Okay, so various safeguards that can prevent infectious disease can be, first of all, proper sanitation of parks, cinema halls, schools, offices, hospitals, and other public places. Next one, a regular cleaning of drainage system and the water reservoirs. Okay, next one is fogging at regular intervals. Vaccination programs, this is very important. Okay, vaccination can prevent a lot of diseases and it can save a lot of lives. Okay, let's continue the discussion. Okay, some of the methods are, first of all, let's talk about the maintenance of the personal hygiene and public hygiene. So this is often the first line of defense against infection. Now, maintaining hygiene is one of the most important ways to prevent infectious diseases. Now, this includes keeping your body clean, eating healthy and nutritious food, and drinking clean water. Now, these habits help your immune system stay strong. Now, public hygiene means things like proper disposal of waste and excreta and regularly cleaning and disinfecting water reservoirs. Now, these steps prevent germs from spreading in the community. Next method is isolation. So this is especially important for disease that spread through air. For example, diseases like pneumonia, chickenpox, and tuberculosis can spread when an infected person coughs or sneezes. Isolation helps stop the chain of transmission. Now, by keeping infected individuals separate from healthy people, we reduce the chances of the disease spreading. Okay, now again, let's continue the answer. Third one is vaccination. Now, vaccination is a powerful tool in a fight against infectious diseases. Vaccination protects the body from communicable diseases by introducing an agent that mimics the microorganism. So we are basically introducing the weakened pathogen or attenuated pathogen or the antigens of that pathogen. Okay. Now our immune system, it responds to this attenuated pathogen or the antigen, and antibodies are created. Okay, antibodies, they develop in our body. And then we have the memory cells. Again, when there is a subsequent infection with the same pathogen, then there is a heightened response in our body and the pathogen is eliminated. Vaccination can also provide passive immunization to the body. This means that sometimes we receive ready-made antibodies giving us immediate but temporary protection. Several vaccines are available against many diseases such as tetanus, polio, measles, mumps, and etc. Next one is vector eradication. This is the fourth major public health measures. Now, diseases like malaria, filariasis, dengue, and chikungunya all are spread by vectors, mainly mosquitoes. Controlling these vectors is essential for disease prevention. Now, we can prevent these diseases by keeping our environment clean and stopping mosquitoes from breeding. Now, one simple but effective step is to avoid letting water stagnate around our homes. Even a small amount of standing water can become mosquito breeding sites. Now, regular cleaning of coolers using mosquito nets and applying insecticides like malathion in drains and ponds are practical measures everyone can take. Introducing fish such as gambusia into ponds is another smart way to control mosquito larvae naturally. Now, this method uses nature to help us fight diseases. Now, let's discuss the next question. The next question is, in which way has the study of biology helped us to control infectious diseases? Okay, so this question is asking, in which way has the study of biology, okay, has helped us to control these diseases? Clear? So there is a lot of research which is being done on the pathogen projection, okay, progression, how the pathogen progresses, how the disease progresses, and how to control it. So keeping this in mind, you have to answer this question. So let's discuss the answer for this question. Now, various advancements in biology have helped us fight infectious diseases. Now, biology has helped us study the life cycles of parasites, pathogens, and vectors, as well as how diseases are transmitted and controlled. Now, vaccination programs against diseases like smallpox, chickenpox, and tuberculosis have helped eradicate or control these illnesses. Now, biotechnology has enabled the development of newer and safe drugs and vaccines. Antibiotics have played a crucial role in treating infectious diseases. Okay, so we can say, to sum up, the study of biology has transformed our ability to control infectious diseases through research, vaccines, biotechnology, and antibiotics. So this knowledge continues to protect us and improve our quality of life. Now let's look at the next question from the NCERT textbook. So the question is, how does the transmission of each of the following disease takes place? Now, understanding how disease is spread help us to prevent them in our daily lives. Okay, so options are, first one is amoebiasis, second is malaria, third is ascariasis, and fourth is pneumonia. So let's have a look at the answer for this question. Now, amoebiasis, this is caused by Antiamoeba histolytica. It is transmitted via contaminated food and water. Okay, so you can see the mode of transmission. It is a vector-borne disease that's spread by the means of contaminated food and water. Now, the vector involved in the transmission of the disease is the housefly. Now, next one, malaria. Now, malaria is spread by the bite of female Anopheles mosquito. The female Anopheles mosquito, this is the vector for this disease. The mosquito transmits Plasmodium parasite to humans during its bites. Now, next one is ascariasis, caused by Ascaris lumbricoides. Ascariasis is transmitted through contaminated food and water. Now, the parasite eggs are ingested with contaminated food or water and they hatch in the human intestine. Next one is pneumonia, caused by Streptococcus pneumonia. Now, this disease spreads through the sputum of an infected person. The bacteria are released into the air when a person coughs or sneezes, and the other person inhales these droplets. So, this is a communicable disease. Okay, infectious disease. Now, next one, What measure would you take to prevent waterborne diseases? Let's discuss this question. So let's have a look at the answer. The waterborne diseases such as cholera, typhoid, and hepatitis B spread by drinking contaminated water. So you can think of contaminated water as a delivery vehicle for harmful microbes. When we drink it, these microbes enter our bodies and can cause illness. Now, these diseases can be prevented by ensuring proper disposal of sewage and excreta and by periodic cleaning. Also, measures such as disinfecting community water, reservoirs, and boiling drinking water should be observed. Disinfecting is like giving the water a protective shield, and boiling is a simple but powerful way to kill most germs before they reach us. Okay, so remember, preventing waterborne diseases is all about breaking the chain of contamination. The next question, discuss with your teacher what does a suitable gene means in the context of DNA vaccines. For more information, you can read NCERT section 7.2. So let's discuss the answer for this question. Now, suitable gene is a specific segment of DNA that, when introduced into a host body cells, can be used to produce specific proteins. Now, this protein kills the specific disease causing organisms in the host body and provides immunity. Okay. Clear? So this is what is known as the suitable gene in the context of DNA vaccines. The next one, name the primary and secondary lymphoid organs. So we are focusing on lymphoid organs here, which are crucial parts of our immune system. So let's discuss the answer for this question. Now, primary lymphoid organs include the bone marrow and the thymus. These are the main sites where lymphocytes are produced and mature. Now, let's look at the secondary lymphoid organs. Examples of secondary lymphoid organs are spleen, lymph nodes, and tonsils, Peyer's patches of the small intestine, and appendix. So, these are the kind of battlefields where immune cells encounter pathogens and mount a defense. Each organ has a specific role in protecting different parts of the body. Okay, in this image, you can see various lymphoid organs. So you can see here the primary lymphoid organ and the secondary lymphoid organ as well. You can see the location of these lymphoid organs. Clear? So you can see here the thymus is shown here, bone marrow is shown here. These are the primary lymphoid organs, and secondary lymphoid organs are also shown. Spleen, you can see the location of spleen, Peyer's patches. Okay, then you can see the lymph nodes here. Tonsils you can see here, and adenoids. These are all secondary lymphoid organ. Moving on to the next question. Now, the following are some well-known abbreviations which have been used in this chapter, Human Health and Disease. You have to expand each one to its full form. Again, you can look at the answer for this question in the NCERT section 7.2 and 7.3. Okay, so abbreviations are MALT, CMI, AIDS, NACO, and HIV. What will be the correct answer? Okay, so let's have a look at the answer. Okay, MALT, this is the mucosa associated lymphoid tissue. Okay, so this tissue acts like a protective barrier at the body entry points, such as the gut and the respiratory tract, helping to stop pathogens before they enter deeper into the body. Now, CMI stands for cell mediated immunity. This is the part of an immune system where cells like T cells directly attack and destroy infected cells. Next one is AIDS. It stands for acquired immunodeficiency syndrome. This is a condition where the immune system becomes very weak, making the body vulnerable to infections that it would normally fight off easily. Then NACO stands for National AIDS Control Organization. So this is the main government body in India responsible for coordinating efforts to prevent and control AIDS across country. Then the last one is HIV, which stands for Human Immunodeficiency Virus. So this is a retrovirus, means it consists of RNA as the genetic material. So this virus attacks the immune system and can lead to AIDS if not managed properly. Here, let's move on to the next question. So next question is differentiate the following and give examples of each. So you have to differentiate between innate and acquired immunity and active and passive immunity. Okay, so let's have a look at the answer. Here, let's talk about innate and acquired immunity. Now you can see here, this table is showing you differences between innate and acquired immunity. So innate immunity is a non-specific, okay? It is non-specific, it is immediate defense, and it is present from the birth, okay? So within innate immunity, skin is involved here, okay, and the cells to fight the pathogens, and it has no memory of the past infections. Whereas acquired immunity, also known as adaptive immunity, is a highly specific response that develops after exposure to an antigen, or it can develop by vaccination also. And it requires days to weeks for activation, but providing long-lasting immunological memory for improved future responses. So there are memory cells in case of acquired immunity. Okay. So here you can see how can we define innate immunity, the natural defense mechanism present at birth, and acquired immunity. This is the immunity developed after exposure to antigens or through vaccinations. Then the response time in innate immunity, this is immediate, okay, minutes to hours. And then in the case of acquired immunity, you have a delayed response, because antibodies, they take time, okay, to develop. So the response is delayed days to even weeks. Then specificity of innate immunity, this is non-specific, and general defense mechanism are involved here, such as skin. Okay. Now acquired immunity, it is highly specific to particular pathogens. Now there is no memory in the case of innate immunity. The response is same each time, and in the case of acquired immunity, it has memory. So response improves with subsequent exposure. So whenever there is subsequent exposure with the same pathogen, then the body generates heightened response, which overwhelm the invading pathogen, and the pathogen is eliminated. Okay, let's have a look at some more differences. So innate immunity is the body's first line of defense, providing a general response against any pathogen. It relies on pre-existing mechanisms like physical barriers, phagocytes, and natural killer cells. Inflammatory response is also part of innate immunity, helping to recruit immune cells to the site of infection. Acquired immunity, also known as adaptive immunity, develops over time through exposure to pathogens. It is more specific, targeting particular pathogens through the production of antibodies by B cells and activation of the T cells as well. So this specificity slows allows for a faster and more effective response upon subsequent exposure to the same pathogen. Okay, this is clear. So here you can see innate immunity. What are the components of innate immunity? Skin, okay, mucous membrane, phagocytes, natural killer cells, complement system, and inflammatory response. In the case of acquired immunity, what are the components? B cells, which produce antibodies, and T cells, both type of T cells, T helper cells and T cytotoxic cells, and memory cells as well. Then duration of innate immunity, this is short term. Acquired immunity, this is long term and can be lifelong. Then let's talk about the evolutionary age. So innate immunity, this is ancient, this is present in all multicellular organisms, but acquired immunity, this is more recent and this is present in vertebrates. So acquired immunity has evolved later. Examples of innate immunity: skin, acid in the stomach, enzymes in saliva, and phagocytotic cells. Then in acquired immunity, you can see examples, or it can be activated through vaccination, okay, recovery from infections, and immunological memory, okay, which leads to heightened response. Let's talk about the active and the passive immunity now. So here active immunity, what is the definition? Active immunity is developed when the body's own immune system produces antibodies in response to exposure to a pathogen or vaccination, while passive immunity is acquired by receiving antibodies from another source. Okay, so ready-made antibodies are being injected into the host. This is known as passive immunity. Active immunity is long-lasting and takes time to develop, whereas passive immunity is short-term and provides immediate protection. Now you can see active immunity, this is triggered by exposure to a pathogen or vaccination. Okay. Now here antibodies are produced by the host itself. The host immune system will produce these antibodies. It is long lasting, often for years or for lifetime as well. Okay, but it takes time to develop this active immunity. Okay, usually it's days to weeks. Now passive immunity, this is triggered by transfer of antibodies from another source. Those antibodies, okay, can be directly injected into the person, such as maternal antibodies. Okay, antibodies present in the colostrum. Okay, they are transferred to the newborn during the breastfeeding. Then through blood transfusions or immunoglobulin injections can also be taken. Antibody injection can also be taken, okay, such as during the snake bite. So here you are directly injecting ready-made antibodies into the host or into any individual. So it is for short term, typically lasting a few weeks only, 2 months. Okay, but it gives immediate protection to the recipient. So these are the difference between active and the passive immunity. Okay, now some more differences between active and the passive immunity. You can see active immunity has memory cells, so it generates memory cells producing long-term immunity. Passive immunity does not generate memory cells, so no long-term immunity. Examples of active immunity are natural infections by the pathogens, vaccination. Okay, for example, vaccines like measles, mumps, rubella, and rubella vaccines. Then passive immunity. Okay, examples: antibodies transferred through the mother. Okay, from the placenta or during the breastfeeding. Okay, and antivenom for snake bites. These also contain antibodies against the, you can say, venom, snake venom. Okay, or snake toxin. Now specificity, active immunity is highly specific to the pathogen or antigen encounter. Passive immunity, this is specific to the antibodies transferred but not as adaptable. Okay, here. So passive immunity, this is specific to the antibodies that are being transferred into the host. So specific antibodies are being transferred, such as against the snake venom. Then active immunity, Okay. What is the booster requirement? So may require booster shot to maintain immunity. In passive immunity, no booster shots are required as the immunity is temporary. Moving on to the next question. So next question is draw a well-labeled diagram of an antibody molecule. So let's have a look at the answer. Okay, antibodies can be represented as H2L2. So this notation tells us that antibody is made up of 2 heavy chains and 2 light chains. Now antibody molecule has 4 polypeptide chains. Okay, the 2 small chains are called light chains. Okay, so there are 2 light chains, that's why L2, and then there are 2 heavy chains, that's why we are calling it as H2. So the molecule is represented as H2L2, which stands for 2 heavy and 2 light chains. Now the antigen binding site is a region on the antibody that attaches to a specific antigen. This antigen binding site is made up of the variable regions of both light and the heavy chains. Now disulfide bonds hold the heavy and the light chains together. Here is a diagram of the antibody molecules. You can notice the Y shape of the antibody, and you can see the heavy and the light chains here, antigen binding sites and the disulfide bonds. You can also see the variable and the constant region in both light and the heavy chains. Okay. Upper one are the variable region, lower one are the constant regions. Now next question, what are the various routes by which transmission of human immunodeficiency virus takes place? Okay, so they are talking about the transmission of HIV. So let's have a look at the answer for this question. AIDS or acquired immunodeficiency syndrome is caused by the human immunodeficiency virus known as HIV. Now, this virus specifically targets the immune system, weakening the body's ability to fight off infections. Now, let's look at the following methods of transmission of HIV. First of all, HIV can be transmitted through unprotected sexual intercourse with an infected person. This means that if someone had sexual intercourse without using protection like condoms, okay, with a person who is having HIV, the virus can pass from one person to another person because these STD, they are spread through body fluids. Now HIV can be transmitted through the transfusion of blood from a healthy person to an infected person. Now by sharing infected needles and syringes, this is another route of transmission. This often happens among people who are using injectable drugs. If a needle is contaminated with HIV infected blood and then used by another person, the virus can enter their bloodstream directly. Lastly, HIV can be transmitted from an infected mother to her child through the placenta during the pregnancy. So this is called vertical transmission. So it's similar to how nutrients and oxygen pass from mother to baby through placenta, but unfortunately, virus can also sometimes cross this wall and reach or infect the fetus. Next question, what is the mechanism by which AIDS virus cause deficiency of immune system of the infected person? Okay. So let's have a look at the answer. Okay, so AIDS, which is acquired immunodeficiency syndrome, is caused by human immunodeficiency virus via sexual or blood-blood contact. Okay, it can be transmitted through sexual intercourse or by blood as well. After entering the human body, the virus attacks and enters into the macrophages. So first of all, they will multiply in macrophages. Now, inside the macrophages, the RNA of the virus replicates with the help of enzyme reverse transcriptase and give rise to viral DNA. So here viral RNA is injected into the macrophages. This RNA is first converted into DNA because the host cell this has DNA as a genetic material. Okay, so when the viral DNA is produced, then this viral DNA is integrated into the host genome. Okay, here so here it's written that viral DNA incorporates into the host DNA and directs the synthesis of new virus particles. Now at the same time, HIV enters helper T lymphocytes. Clear? So this is the critical entry point where the virus begins its attack on the immune system. So T lymphocytes are being infected here as well. Now again the HIV virus will multiply. Okay, then it will burst the T lymphocyte cell. So slowly the number of T lymphocytes starts decreasing. Clear? So now this virus replicates and produces viral progeny within the T lymphocytes. These newly formed progeny viruses get released into the blood, attacking other healthy helper T lymphocytes in the body. As the number of T lymphocytes in the body of an infected person decrease progressively, thereby decreasing the immunity of a person. And this is the main reason why people with AIDS are so vulnerable to infections. Okay, so let's discuss next questions. How is cancerous cell different from normal cell? So let's have a look at the answer. Okay, here you can see this is a table showing a difference between normal and the cancerous cell. Now, normal cells, what is the growth rate? Controlled. Normal cells, their growth is controlled, it is regulated, where cancerous cells, they show uncontrolled growth and the growth is quite rapid as well. Now, in normal cells, okay, the cell cycle is regulated. So, there are certain checkpoints, clear, which are strictly regulated. In cell cycle, there are certain checkpoints. These checkpoints are strictly regulated in case of normal cells, whereas in the case of cancerous cells, these checkpoints are often bypassed or ignored. Then these normal cells, they undergo programmed cell death if they are damaged, also known as apoptosis, and cancerous cells, they evade apoptosis, leading to survival of damaged cells as well. Then normal cells, they fully differentiate and specialize, whereas cancer cells, they are poorly differentiated and often lose specialized functions. Now normal cells, they have the or they show the property of contact inhibition, means they stop dividing when in contact with other cells, whereas cancer cells, they lack contact inhibition and they continue to grow and pile up, forming tumors. Okay, some more differences. So here you can see in normal cells, the genetic stability, this is relatively stable. They have relatively stable genome. So normal cell maintain a relatively stable genome, while cancerous cell exhibit high genetic instability with frequent mutations. Okay. Now let's talk about angiogenesis. Now normal cells undergo angiogenesis to supply developing tissues with oxygen and nutrients, but cancer cells actively induce the formation of new blood vessels to support their rapid growth and spread. Let's talk about metastasis. So normal cells do not spread to other parts of the body, while cancer cells can invade nearby tissues and migrate to distant organs, establishing new tumors. Let's talk about the energy production. Normal cells primarily use oxidative phosphorylation for energy production, while cancer cells often rely heavily on glycolysis, even in the absence of oxygen. A less efficient process, but one that allows for rapid cell growth. Okay, let's continue the discussion. Now here you can see the normal cells. They respond appropriately to growth signals, whereas cancerous cells, they often grow in the absence of growth signals or produce their own growth signals. Now, normal cells, let's talk about normal cells, immune system interaction, recognized and often eliminated by the immune system. And cancerous cells, they can evade immune detection and destruction. That's why they keep on growing and multiplying. Now, these differences are fundamental to understanding how cancer develops and progresses, and they also form the basis for many cancer treatments and research strategies. The next question, explain what is meant by metastasis. So let's have a look at the answer. Now, the property of metastasis is exhibited by malignant tumors. Now, metastasis is the process where cancerous cells spread to different parts of the body, and this is the key feature that makes cancer so dangerous, as it allows the disease to move beyond its original location. Now, these cancer cells divide uncontrollably, forming a mass called a tumor. Now, some of these tumor cells break away and enter the bloodstream. Now, the blood acts like a transportation system, carrying these cells to other parts of the body, much like a river carrying seeds downstream. Once in the bloodstream, these cells can reach distant organs and start forming new tumors by dividing actively. Now, this is why metastasis leads to development of secondary tumors, making cancer treatment more complex. Now moving on to the next question, list the harmful effects caused by alcohol or drug abuse. Let's have a look at the answer. Now let's talk about the physical effects first, physical effect of alcohol and drug abuse. Now, alcohol and drug abuse can have several ill effects. First, these substances can affect the body of the individual. This means damage to organs like liver, heart, and brain. Now, there can be serious health implications, even death. Overdoses, accidents, and long-term diseases are all possible outcomes. Drugs and alcohol also affect the fetus in case of pregnant women. The developing baby is especially vulnerable, and exposure can lead to birth defects or developmental delays. Now, these things also have a bad influence on families. Substance abuse or drug and alcohol abuse often leads to arguments, mistrust, and emotional pain. The family, which should be a support system, can become a source of stress. Now, consumption of excessive alcohol devastates the family and leads to several domestic problems. This can include financial issues, violence, and breakdown of relationships. Now, it also causes lack of interest in social activities. People may withdraw from friends, hobbies, and community events. Now, this isolation can make recovery even harder. Alcohol and drug lead to criminal activities too. Sometimes people may steal or break the law to support their addiction, or their judgment is impaired, leading to risky behavior. Drugs also accelerate the chance for spread of infections like HIV, sharing needles, or engaging in unsafe practices, increasing the risk of serious diseases. Moving on to the next question, do you think that friends can influence one to take alcohol and drug? If yes, how may one protect himself or herself from such an influence? Okay, let's discuss this answer. Yes, friends can influence someone to take drugs and alcohol. Peer pressure is a powerful force, especially during adolescence. Okay, now let's look at the steps a person can take to protect themselves from drug abuse. First step is to increase your willpower to stay away from alcohol and drugs. Next, avoid experimenting with alcohol just out of curiosity or for fun. sometimes what start as a small experiment can lead to bigger problems. It's better to stay safe and not take unnecessary risk. Another important step is to avoid the company of friends who take drugs, just like you avoid unhealthy food to stay fit. Avoiding negative influences help you stay fit and healthy. Now, if you feel pressured, seek help from your parents and peers. Talking to someone you trust can give you the support and advice you need to make the right decision. Now, take proper knowledge and counseling about drug abuse. Understanding the harmful effects of drugs can help you make informed choices and avoid dangerous situations. Devote your energy to extracurricular activities. So, engaging in sports, arts, or hobbies keeps us, or this will keep the person busy in a positive way and reduces the chance of falling into bad habits. Now, if you notice symptoms of depression or frustration, seek immediate professional and medical help from psychologists and psychiatrists. And mental health is just as important as physical health, and getting help early can make a big difference. Okay, next question. Why is that once a person start taking drugs and alcohols, it is difficult to get rid of this habit? You have to discuss it with your teacher. Okay, so let's have a look at the answer. Now, drug and alcohol consumption is addictive because it creates a feeling of euphoria and temporary well-being. Now, with repeated use, the body's receptors become less sensitive, so the person needs more of the substance to feel the same effect. So this is called as tolerance. Moving on to the next question, in your view, what motivates youngsters to take alcohol or drugs, and how can this be avoided? So let's have a look at the answer. See, the main motivating factor that stimulates youngsters to take alcohol or drugs are: the first factor is peer pressure. So this is when friends or classmates encourage someone to try alcohol or drugs, often making it hard to say no because everyone wants to fit in. Next is the desire for excitement. Some young people are looking for new experiences or thrills, and they might see alcohol or drugs as a way to add excitement to their lives. Another reason is liking of taste. Sometimes the taste of certain drinks or substances can be appealing, especially if others talk about it positively. Now, some youngsters use substances to escape from frustrations. When life feels overwhelming or stressful, they might turn to alcohol or drug as a way to temporarily forget their problems. The family history is also important. If substance use is common in the family, okay, means if the drug and alcohol abuse is common in the family, youngsters may be more likely to use it. Here, now, next one, curiosity due to advertisement. So ads can make alcohol or drugs look attractive or glamorous, which can spark curiosity in young people to try them. The next one is false beliefs of enhanced performance. So sometimes, okay, these drugs they give you, you can say feel of, you can say pleasure, and you can say energy, but these are some false beliefs. So some people think they can enhance your performance, they can enhance your energy, okay, they can give you feeling of pleasure, but that is a false belief. Actually, they are dangerous for your health. The next one, finally, youngsters use substances to overcome the hardship of daily life. Now, when facing challenges, they might look for an escape, even if it's only temporary and harmful in long run. Okay, now next one, alcohol or drug abuse can be avoided by. So it's important to identify the reason for addiction and you have to take remedial measures. Okay, next, avoiding undue peer pressure is The key: peer pressure can be like a strong current in a river pulling you along even if you don't want it. Now, proper education and counseling are powerful tools. Okay, seeking help from parents, teachers, and trusted friends can also help avoid alcohol and drug abuse. Again, seeking professional and medical help will also help to prevent alcohol and drug abuse. Okay, so this was the end of the session. I hope the session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.